Hello, my name is Alba Mawala, and today I'll be presenting my research titled Effects of Dietary Sugar on Reproduction in Drosophila melanogaster. In this project, we studied how different amounts of sugar in the diet affect reproductive success in fruit flies and how these findings relate to metabolism and human health. Why we study nutrition and why we use fruit flies? We study nutrition because it plays a major role in growth, metabolism, and reproduction, and sugar are an important energy source for biological process. We use fruit flies or Drosophila melanogaster because they are commonly used in biological research since they reproduce quickly, are easy to maintain, and share many conserved metabolic and genetic pathways with humans. Because of this, fruit flies are an excellent model for studying how diet affects biological processes like reproduction. The main research question of this study was how does dietary sugar concentration affect reproductive success in fruit flies? The hypothesis: median sugar levels will produce the highest reproductive success. Very low sugar would limit reproduction due to lack of energy, and very high sugar would reduce reproduction due to metabolic stress. The experimental design. To test this, we prepare five different sugar treatments. We prepare five vials with different sugar concentration levels, such as 0%, 1.25%, 2.25%, 3.25%, and 4.25%, to determine which percent is the optimal medium for the development and reproduction of fruit flies. All other conditions were kept constant, including yeast and water content. We add five male and five female flies to each vial, and the reproductive success was measured by counting pupa daily from day 7 to day 12. And the pupal count was used as an indicator of successful reproduction and development. The results. This graph showed pupil development over time. In the early days, development was slow in all groups. However, after day 9, the 3.25% sugar groups show a rapid increase in pupil formation. This suggests that the sugar concentration provide optimal conditions for development. The other groups developed more slowly, especially at the very low and very high sugar levels. The final reproductive output. When looking at final pupil counts, the difference becomes clear. The 3.25% sugar group produced the highest number of pupa with 91 total. The 0% sugar group produced the fewest with 41 pupa. And the highest sugar group, 4.25%, showed reduced reproduction with 68 pupa. To explain these results, I connect my finding to previous research that we studied during classes. One of them is by Mondonori and other authors. They study how diet affects gene expressions in fruit flies. They identified 228 genes that change expression depending on diet, especially genes that involve in mitochondrial function and energy metabolism. This show that diet directly affects how cells produce and use energy. Um, in the Montanori study, uh, we can visualize gene expression chains. For this, we use this heat map, where red colors indicate higher gene expressions and green colors indicate lower expression. Different diets produce distinct gene expression patterns, particularly affecting mitochondrial and metabolic pathways. These molecular changes help explain why high sugar intake disrupts energy metabolism and reduces reproductive success, as seen in my experiment. Another interesting author, it's Muselman and other authors, and they show that high sugar diets cause metabolic dysfunction in flies, including resistance that it would lead to diabetes. It's important to know that about 75% of human disease related genes have equivalents in fruit flies. So we can infer that we can use flies to study and predict possible disease in humans. These findings are relevant to human health because fly and humans are similar biological systems to regulate sugar metabolisms. When flies consume excessive sugar, these systems become disrupted, leading to the metabolic imbalance. And this highlights why balanced nutrition is essential for reproductive and overall health across species. To summarize, we create this conceptual model that allows us to summarize the pattern seen in the results. And at low sugar levels, flies don't have enough energy for egg production and larval development. So the reproduction levels are low. And at optimal sugar levels, energy availability is balanced and metabolism work efficiently and reproduction is maximized. And at high sugar levels, metabolic imbalance and stress reduce the reproduction efficiency. In conclusion, this study showed that a sugar concentration of 3.25% maximized reproductive success in fruit flies, while both low and high sugar levels reduced reproduction. These results align with the molecular evidence from previous research and support the importance of a balanced nutrition. Thank you for watching my video and feel free to ask any question